In 1859, Charles Darwin published a book titled On the Origin of Species. The pages described natural selection as a slow mechanism by which living things gradually change across countless generations. The work rested on decades of patient observation gathered during calm sea voyages and long walks through damp English countryside. The ideas settled into the scientific world quietly, like dust motes drifting through a narrow sunbeam in an otherwise still room. In 1865, Gregor Mendel presented his experiments with pea plant hybridization at a Natural History Society meeting in Brno. He had spent 8 years in his monastery garden recording the height of stems, the color of seeds, and the texture of coats through thousands of quiet observations. His findings established the fundamental laws of inheritance. The same ratios appeared again and again, steady and predictable, revealed only to a monk with enough patience to tend each row through gentle rain and summer warmth. In 1869, the Swiss chemist Friedrich Miescher isolated a substance from the cell nuclei of white blood cells. He extracted it from pus collected on used hospital bandages in a still room near the Rhine, and he named the unfamiliar material nuclein. This pale, slightly sticky substance was the first crude isolation of DNA. Miescher simply noted its presence with the calm precision of a man weighing salt or filtering water, not yet knowing what it carried inside each microscopic strand. By the close of the 19th century, these three threads remained separate and largely unnoticed by one another. Darwin's natural selection, Mendel's laws of inheritance, and Miescher's nuclein each rested in its own quiet corner of the scientific world, waiting in bound journals and still laboratory notebooks. The conceptual and chemical foundations of heredity had been laid down with patience, care, and no sense of urgency. Decades would pass before anyone understood how closely the three discoveries were meant to be woven together. In the year 1900, 3 botanists working in separate countries opened decades-old copies of a local natural history journal. Hugo de Vries in Holland, Karl Correns in Germany, and Erich von T. Schermack in Austria each found Gregor Mendel's 1866 paper without knowing the others had done the same. They read his careful ratios of tall and short pea plants, and each man slowly recognized that the same laws were appearing in his own breeding experiments with corn, peas, and other flowers. The rediscovery passed like a soft ripple through botanical circles, linking isolated laboratories with nothing more than paper and ink. In 1902, a young American biologist named Walter Sutton spent long hours studying the cells of a grasshopper under gentle lamplight. He watched the dark chromosomes pair along the center of each dividing cell, then separate cleanly into 2 new cells in a process called meiosis. The pattern was unmistakable. The chromosomes split into matching pairs in precisely the same numerical ratios that Mendel had found in his peas, as though the invisible laws of inheritance had suddenly become visible under the lens. By 1911, Thomas Hunt Morgan and his students at Columbia University had spent years breeding generations of fruit flies in quiet rooms filled with the faint smell of yeast and banana. They tracked thousands of flies with white eyes, vestigial wings, and bristled bodies, recording each birth in thick, penciled notebooks. The flies multiplied in their bottles, and the students counted them by lamplight, filling page after page with calm, steady observation. Their work showed that genes travel on chromosomes, and that some traits are inherited together more often than others in a pattern they called genetic linkage, binding the physical thread of the chromosome to the abstract unit of heredity. Years before the fly rooms filled with counting and lamplight, Archibald Garrod noticed that the urine of certain patients darkened to a deep brown-black after sitting quietly in open air. He followed this condition, which he named alkaptonuria, through the branches of family pedigrees and saw it obey the same ratios Mendel had found in his pea beds. The illness appeared only when two hidden factors came together in one child, proving that human disease could follow the quiet rules of heredity. Garrod filled his pages with these lineages, connecting body chemistry to invisible units traveling unchanged from parent to child. In 1909, Wilhelm Johannsen gave the hidden factor its lasting name. He called it the gene, a small word for the unit of heredity that existed apart from the living form it shaped. He drew a careful line between genotype, the inner inherited nature, and phenotype, the outward appearance of body or seed. The distinctions settled into scientific speech slowly, like cool water filling a deep vessel, offering a language to separate blueprint from building. The new terms spread through journals and lecture halls, with the same patient rhythm as the beans growing in his Copenhagen plots. By 1941, George Beadle and Edward Totham tended colonies of Neurospora crassa in tall glass vessels, offering the mold minimal diets and observing which strains failed to grow without specific vitamins. From thousands of these quiet omissions, they proposed that one gene directed the making of one enzyme. The one gene, one enzyme hypothesis drew a straight path from the abstract unit to the chemistry that sustains life. Their pink colonies grew in steady silence, bridging the gap between the invisible gene and the tangible substances that build a living cell. The work completed a thread that began in Garrod's marble clinic and Johannsen's bean-filled study, binding name to function in a single calm arc. The same years brought a quieter question about the physical substance of genes. In a New York laboratory, Oswald Avery, Colin MacLeod, and Macklin McCarty prepared gentle extracts from heat-killed bacteria and mixed them with living cells. They watched the colonies transform slowly in glass Petri dishes, one strain becoming another. Their painstaking tests showed that DNA alone carried the transforming power, while proteins held none of it. Across the country, Barbara McClintock studied the speckled colors of corn kernels under lamplight. She traced the mottled patterns to genes that moved from one chromosomal site to another in quiet motion. These transposable elements slipped through the genome like loose threads shifting in a woven cloth. Her 1944 discovery revealed that inheritance was not fixed in place, but could rearrange itself within the living architecture of the cell. 8 years later, Alfred Hershey and Martha Chase used a simple kitchen blender to shear phage coats away from infected bacteria. They found that viral DNA entered the cells while protein coats remained outside in the spinning liquid. The blender hummed with a steady soft whir, confirming that DNA alone directed the infection. Their experiment removed the broad remaining doubt that protein might carry hereditary information. These separate threads converged in the middle of the century. DNA was recognized as the stable hereditary material, yet McClintock's jumping genes showed it was also fluid and alive with quiet motion. The genome could shift and settle into new patterns over seasons and generations, rewriting its own map in slow increments. A steady picture of life began to form from these deliberate observations in cornfields and chilled laboratories, one calm finding laid gently upon another. In a quiet basement laboratory in London, Rosalind Franklin adjusted the camera that held her precious DNA fibers in a humid flow of hydrogen gas. She worked with refined X-ray diffraction techniques, letting a fine beam of light pass through the glassy threads and scatter onto photographic paper tucked in the dark. The exposure took many hours, and the air smelled faintly of photographic fixer. In 1952, one of these plates, later known as Photograph 51, developed into a clear cross-shaped pattern of dots that suggested a helical shape turning gently in space. At Cambridge, James Watson and Francis Crick stood at a wooden table and fitted small metal plates and wire spokes into possible molecular forms. They measured the angles and distances from Franklin's diffraction images and recalled the numerical pairings of Chargaff's rules: adenine with thymine, guanine with cytosine. In 1953, they described the double helix, two strands winding around each other in a slow ascending spiral joined by rungs of paired bases. The structure could unzip and copy hereditary information with quiet precision. Across the North Sea in Lund, Sweden, cytogeneticist Johin Tgio treated human cell samples with a diluted salt solution that swelled the nuclei and spread the chromosomes wide apart on the glass slide. He looked through the eyepiece of a brass microscope and counted the distinct, paired bodies one by one against a pale field of light. In 1956, he established that the correct number of human chromosomes in each cell is exactly 46, settling decades of uncertain estimates that had ranged far higher and lower. The double helix and the exact chromosome count arrived within a few quiet years of one another. These findings gave physical shape and definite number to the material that McClintock had shown could move and rearrange. A solid foundation had been prepared from patient measurements and long exposures. The geometry of inheritance now rested in plain view, steady and orderly, waiting for the next calm observation to arrive in its own time. The geometry of inheritance now rested in plain view, steady and orderly, waiting for the next calm observation to arrive in its own time. In 1961, Marshall Nirenberg sat in a quiet room at the National Institutes of Health and arranged chilled glass beakers on a wooden bench. He mixed synthetic RNA with cell extracts and watched as amino acids linked together in ordered chains. This was the genetic code made visible, the exact instructions that translate genetic material into protein synthesis. Each triplet of bases corresponded to a single amino acid, spelling out the language shared by nearly all living things. 16 years later, in 1977, Frederick Sanger developed a rapid DNA sequencing technique that read the order of nucleotides directly. He used chain-terminating nucleotides and gel electrophoresis to separate fragments by size. The dark bands on film and the printed peaks on paper revealed the exact sequence of letters within a gene. For the first time, a scientist could read a paragraph of heredity as easily as tracing a line with a finger. Knowing the sequence was only the beginning. A single drop of blood or a root hair contained vanishingly small amounts of DNA, too faint for repeated study. Scientists needed a way to duplicate the fragile strands without growing entire colonies of bacteria. The problem sat on the bench in the form of scarcity, a simple lack of sufficient material for the questions at hand. In 1983, the polymerase chain reaction, known as PCR, was invented, allowing scientists to amplify DNA. A mixture of primers, polymerase, and nucleotides cycled through gentle heating and cooling in a modest machine. Millions of identical copies accumulated in the small plastic vessels, turning invisible traces into material that could be held and studied. The method gave researchers the ability to multiply a fragment of genetic text until it filled the page, steady and readable in the soft light. The invention of PCR in 1983 gave researchers the means to multiply DNA fragments with patient repetition. That same year, Using these amplified signals as guides, scientists mapped Huntington's disease to its chromosomal location. The disorder became the first genetic illness traced to a specific site, a narrow band on the short arm of chromosome 4. It appeared as a soft gray stripe on a stained slide, visible beneath the objective lens in the stillness of the late afternoon lab. 6 years later, in 1989, the gene mutation responsible for cystic fibrosis was finally identified. The search had narrowed to a region on chromosome 7, where a precise deletion of 3 nucleotides produced a defective protein. This single small loss disrupted salt transport in the lungs and pancreas. The finding explained the chronic infections and digestive failures that had been recorded in patient folders for generations, and it settled into the files with the quiet weight of a solved mystery. The Human Genome Project officially began in 1990 with international agreements and the steady allocation of funds for large-scale sequencing. That same year, researchers provided the first evidence for the BRCA1 gene, linking a region on chromosome 17 to hereditary breast cancer. The two undertakings promised to fill in the map base by base. Anonymous chromosomal landscapes would slowly become named neighborhoods where disease could be located and studied in the soft light of accumulated knowledge. The decades of searching had produced a set of reliable methods and a handful of known addresses on the chromosomes. Researchers arrived each morning to carry the work forward in the same calm manner, repeating experiments in the soft hum of cooled rooms. The genetic map was no longer a blank page. It had become a quiet atlas of specific locations, each one marked with patience and read by the steady eye. In 1995, scientists read the complete genome of Haemophilus influenzae from end to end. The bacterium's single circular chromosome held roughly 1.8 million base pairs, traced in the soft glow of monitors after months of automated reading. It became the first free-living organism whose entire genetic text was spelled out without gaps. The achievement rested on years of methodical work in cooled rooms where machines clicked through samples with steady rhythm and soft whirring. Chromosome 22 was the first human chromosome to be fully decoded. In 1999, its 48 million base pairs were arranged in order after patient collaboration across many laboratories on 2 continents. The smallest human autosome had surrendered its complete sequence, revealing the dense architecture of heredity in a form that could be read base by base. The data arrived as long, silent strings of letters, filling screens in the early morning hours while printers released thin, warm sheets of sequence. The first draft of the human genome appeared in 2001. Printed in scientific journals with understated type, it covered the great majority of gene-containing regions across 23 pairs of chromosomes. Researchers turned the pages slowly, finding thousands of genes nestled among long stretches of quieter, repetitive sequence. Colleagues shared the heavy bound volumes in hushed reading rooms, noting the blank spaces that remained calmly marked for later finishing. The Human Genome Project was declared complete in 2003. The reference sequence exceeded 3 billion base pairs, a finished atlas assembled from many hands working over more than a decade. Soon after, next-generation sequencing machines began to hum in laboratories around the world, reading DNA with a speed that once seemed distant. Automated analyzers processed millions of fragments in parallel under soft blue light, while the accumulated knowledge settled into databases like still water, waiting to be touched